Welcome to the autumn series on light. This is the 12th of our online series of lectures. All the others are on our website and on YouTube, and nearly 70 hours worth of quantum biology lectures there. And I'm going to give a slightly longer introduction today because we've got quite a lot of new participants on the meeting today, and the number seems to be going up all the time. So many of you know me. I'm Geoffrey Guy, the founder and chairman of the Guy Foundation, and I established the foundation with my wife Kate in 2018. And main idea was to promote the study and the research of quantum effects in biology, and probably with special reference to bioenergetics. And my partner in that, the scientific partner in that endeavour from the early days was Alistair Nunn, our director of science. All of you will have had contact with Nina at some point. Nina's our program director, and Bethany Adams, who is in South Africa, looks after our scientific communication. So and it's interesting that Alistair and I really started thinking about quantum biology when we started to see some rather odd effects of photons in Stan's laboratory when we were doing some microscopy on cannabidiol. And then we found there were other effects on other chromophores. So it's taken us about, what are we saying, 8 years now to come full circle back to this series on light, which I think is very exciting. The last series, of course, was on water, and that was a real eye-opener for a lot of people, actually. And I think light is absolutely the next step for us. So I would like to introduce a very good friend of the foundation, Dr. Michal Cifra. Michael is head of Bioelectrodynamics research team and a senior scientist at the Institute of Photonics from the Czech Academy of Sciences. After obtaining his PhD in Radioelectronics from the Czech Technical University in Prague, Michal undertook research at institutions in both Germany and the United States before taking up his post back at the Czech Academy. His research interests focus on active and passive electromagnetic properties of biosystems, and in engineering EM field biomatter interactions at the nanoscale, particularly focusing on proteins. Michael's a very valued member of our scientific faculty, and he's also known for his role in editing some key quantum biology books. Many of you will know Fields of the Cell, and one I can recommend very strongly, Ultra-Weak Photon Emission from Biological Systems. Okay, so Michael, without further ado, can I hand over to you for to present your lecture?
Yes, thank you very much. Okay, thank you very much for such a wonderful introduction. Indeed, I'm really enjoying this series. I'm glad that I can contribute with kickstarting this wonderful series of Life and Light. And this is really an introduction, a brief one, of the physics of photobiology. So this is our team, was already briefly introduced. We are people ranging from engineering to biophysics and physical chemistry, located here in Prague, in Czech Republic. And we are really dealing with the interaction between the electromagnetic light and the biology. So what is light? By definition, it's actually both electromagnetic wave and all composed of particles. Just two classical and quantum physics perspectives are valid for light. It can also be called wave, radiation, or field and oscillations. What are important quantities is the frequency, then inverse of it through the speed of light is the wavelength, and it's just a really small part of the spectrum. And as you can see here, it's really a very small part of the electromagnetic spectrum, but very special for us. And what are other important quantities are also the energy of individual photons. That matters a lot, because these are what is often used in physics and biophysics to determine whether energies of this is conceptually then coupled to the energies of the states and electrons in the molecules which absorb light. To expand this a bit, well, just to keep us on the same page, so this is the standard convention of the colors as we perceive them by the human eye, and related to the wavelength and the frequencies and the photon energy. I consider it quite useful table. So we are speaking of hundreds of terahertz of the frequency, hundreds of nanometers in wavelength, and about the few units of electron volts. So why is light essential for life? you will see next slide. So it plays fundamental role in life processes. Obviously, all the evolution and energy supply from in our star system, it's provided by the sun to the Earth. So this is powering the life both from energy and also from entropy perspective. Well, light enables plants to grow, then to be consumed by the animals. So the energy is propagated through the whole ecosystem and food chain. And well, the first, if we go the closest to us, to humans, is absolutely important in terms of being able to use our sight. So next slide, please. So I would like to start from a biophysics perspective, giving a molecular perspective on the light biology interaction. So what absorbs light on the level of the biological systems? The most classic understanding, it is the chromophores. The most simple one are those which are these are the bonds between the carbons, or also can be between other atoms, nitrogen, for example, where the double bonds, so 2 electrons shared, are alternated with the single bond, 1 electron shared. And if this is alternating, this is a perfect structure to absorb light. Often in nature, we can see these structures in multiple repetitions or in cycles, like these benzene ring. Next, please. So very abundant chromophores are present, for example, in aromatic amino acids, and there are aromatic amino acid residues in proteins. Most effective absorber of light, however, in a near in ultraviolet is tryptophan, then it's tyrosine and phenylalanine, but by far, absolutely, it's the tryptophan and tyrosines which win the game in absorbing most effectively. Then you can see here the spectrum of other important light absorbers in organisms, which is denucleic acid, or also RNA and hemoglobin and melanin are very important absorbers in tissues of mammals. And what is interesting here, that somehow the water itself is also a good absorber in near infrared, but just in the visible range there is kind of a water window for lower absorption, so that the light can penetrate deeper to the tissues. Another important class of light absorbers are opsins and cryptochromes, which have often been mentioned in the context in quantum biology, especially in magnetosensing. So you can see here that these kind of molecular species cover really the whole visible range and beyond. And importantly, what seems to be very important for light effects in biology and in the photobiomodulation therapies are cytochromes. And you can see these absorption spectra of a variety of cytochromes, and also for comparison, the hemoglobins here in this graph. So they are ranging really from visible range up to the near infrared, where there is a broad peak of cytochromes. So I'd like to explain what happens on a really fundamental level with the photon after it's absorbed by the molecule. So we have to look on the scale of the electron. So imagine before and light comes in, our system, be the molecular system, is in a ground state. Then when photon comes, there is an absorption, and the electron from the ground state is excited by the energy photon to go to the first excited state. It has to be of the same spin, because the spin can be changed in this transition. So it's still the same orientation of the spin of the electron on the higher excited state. So it's so-called singlet state. And it can be excited to variety of sub levels. So these thinner lines are the vibrational sublevels of particle electronic state. So S0 is the electronic state, S1 is the ground state, S1 is excited state, and these thin lines are vibrational sublevels. Then what happens very fast, much faster than anything else usually, is vibrational decay. So electron with this state decays to the ground state of vibrational state very fast. So this is also can trigger vibrations of the whole structure, or release as a heat. Then what can also happen, the absorption also can happen to the higher excited states. Then what can happen is that from this higher excited state, what's happening is the transition to the lower excited state. And the first important thing, which is often employed in the variety of imaging techniques and detection techniques, is the decay of the excited state, of the electronic excited state to the ground state, while emitting light of the longer wavelength. And if there happens to be the same spin, so it's single to single transition, it's so-called fluorescence on a scale of nanoseconds or tens of nanoseconds. Another phenomenon which can happen, and it's very important for quantum biology, is transition from the single state to triplet state, still electron excited states, but only difference is the spin changes, and often the spin of the electron changes. Often these triplet states are lower lying, and so-called intersystem crossing is the process in which the electrons transits from the single to the triplet state. And often this transition, which can be bidirectional, which is often influenced by the magnetic fields in the molecules. Then once the electron is in the triplet state, and if the transition to single state occurs, which is quantum mechanically forbidden, but there are tricks of how the nature can bypass this forbidden transition, which effectively means it takes longer to happen. This is so-called phosphorescence, which can take from microseconds to even tens of the seconds. What is interesting here is that excited states do not just manifest interesting physics. They can also manifest interesting chemistry. Excited states of the electrons are very chemically reactive. They can lead to transfer of electrons. So excited states, electron, it's easier to transfer to other molecules. They can cause scission of the bond, so the molecule can fall apart, or they're much more reactive, so they can react with other molecules or other molecular groups within the same molecule. What is interesting is also that excited states doesn't have to be just stay on that particle part of the molecule or in that particle molecule. It can be transferred via so-called near field energy transfer or Foster resonant energy transfer, FRET. So the excited state of the donor molecule can transfer its excitation energy to the acceptor. This is different from just having photon emitted and caught by some other molecule somewhere elsewhere. This transfer can be extremely effective, is really distance dependent, and is a really so-called near field transfer. So it's not emitted light energy. The energy was not emitted as a photon, it's still just kind of near photon field at the atom. So after light is absorbed and all these phenomena can happen, what is the biological response? I will be very concise here because we're going to hear all about this through all the series, I believe. So what is known about, I would say, most general mechanisms which are pretty much applicable to most of the eukaryotic biology is that as soon as you apply higher energy light, speaking of bluish, violetish part of light, this light is going to excite endogenous photosensitizers. That means the molecules which are easy to excite, and those can transfer these excitation energy most often to oxygen, to excite it, to create single oxygen. And this is the starting point to generate a variety of reactive oxygen species, including hydrogen peroxide and other longer living reactive species, which then can trigger biological response. And often to excite these photosensitizers, the chromophores, which can generate ROS further, for this, most of the time, one needs to have a higher energy light, so therefore blue. At the reddish part of the spectrum, it seems to be, from the literature, from great work of Mike Hamlin and others, and Tina Caro, and many other researchers, it seems to be that the mitochondrial modulation is the key to these mechanisms. So red and near infrared light seems to be the wavelengths which can modulate the function of electron transport chain, especially in the complex 4, which contains the cytochromes. And these then, in a downstream cascade, can lead to a change of the mitochondrial transmembrane potential, then, of course, to ATP generation, because transmembrane potential is powering the generation of the ATP in classical bioenergetics paradigm. And also this can affect signaling of nitric oxide. Then there are many others very interesting but cell-specific responses, which are really not present in all of the cells, but only in specific cells, which, for example, contain opsins or modification of those in part of the membrane channel. So certain type of the cells can either natively have the function or can be engineered to be sensitive to light by changing the permeability of certain ion channels. So these actually something that can be used to manipulate the membrane potential with light. So I also like to take in a bit of structural perspective. I would say more physical and macroscopic perspective. So far, I've mentioned only the case what happens if the photon impinges a molecule, which is when it's a good absorber, it absorbs the light and excites the electron to excited states, which can have many fates. However, structurally, physically, there are more other interesting complex phenomena which are happening with light. Obviously, from the high school's physics, is transmission through the interface, reflection on the interface, refraction, diffraction, absorption we mentioned, or scattering. So the light changes direction. This can lead to interesting phenomena. Consider that the biological structures are quite rich, but they have certain patterns. For example, I developed a toy model some 13 years ago, starting after my postdoc, actually the postdoc time, and I developed a model where I was thinking, okay, let's look on a structure of the spherical model of the cell, and look where its nucleus is typically located, usually in the center. So classical centering mechanisms take into account the forces of the cytoskeleton, which mechanically guide the nucleus to the center. However, when the cell divides, there has to be also division of the newly formed nuclei. So, and I just found coincidental shapes which are similar to modes of the cell as if they are considered a dielectric resonator or a light resonator, if you like. So I just took the size and properties of the cell, calculated what are its electromagnetic eigenmodes, and it come up with the solution that it actually happens to be in a mid-IR and the higher modes than there are in a let's say visible range of light. So somehow you could speculate that some contributor to the organization of the organelles in the cells could be partially also electromagnetic. But this is, I would say, still speculative, just an interesting coincidence in the shapes. What is also interesting that we, of course, in a higher organisms with differently shaped fibers and structures, including blood vessels and nerves, which again have interesting optical properties in the sense that they potentially as a structures with certain refractive index can guide light. Just based on a total internal reflection along these structures. So there are actually multiple papers starting from 70s up to the really current ones, as this one from Zeng in applied optics, where the authors analyzed what are the propagation modes of the light along for example, the nerves, the neurons and axons. So this is actually especially important in the context that biosystems are also themselves source of photons of light. And this is what is my expertise, especially in our research team. Okay, so we know very well in textbooks that electromagnetic field is generated by higher organisms, even by the cells in a low frequency spectrum. Basically, membrane fluctuations are all within few kilohertz. Then there is kind of terra incognita in the radio frequency and microwave range, where it's not much known about endogenous electromagnetic signals. And then there is a light side in the non-ionizing part of the electromagnetic spectrum, where it is well known that there are specialized organisms which can shine light so that you can see it by naked eye, but it's a little bit less known that basically all biomaterial shine light even without the presence of these specialized enzymatic pathways which can generate very bright light. And these ubiquitous indigenous light, we call it biological autochemiluminescence. So we define this phenomenon as a luminescence from biosystems where electron excited states are formed due to the energy from oxidation of endogenous biomolecules. So in other words, it's the indigenous metabolism and biochemistry which generates in dark, even without presence of external light, excited states. And this is very important because excited states have then very rich physics and biochemistry. Well, what is known experimentally is that this phenomenon is observable very well in these visible and near infrared range, with some reports going into the near UV, which is very interesting. This phenomenon is very ubiquitous, as I mentioned. Any biomaterial will demonstrate this phenomenon, especially the living one will have it also controlled based on the metabolism. And And the intensity, however, is pretty low, and it's about 1,000 of photons per centimeter squared per second across the whole visible range. There are a variety of phenomena or synonyms for this in literature, so you can search more wider. I think I'm showing here my favorite depictions of how this works. So once we have the reactions of the biomolecule and oxidant, this with some intermediate steps lead to excited state product. This is the P with the asterisk. It means the electron is excited to the higher level. And then what can happen is one of the fates is that it's a radiative transition where we should see in animation there is a photon coming from this. Anyway, yeah, that's basically this how the photons are generated by endogenous biological chemistry. Well, we can detect these signals. We can even image them with sensitive cameras, and that's what our lab has great expertise in. Well, we can either do images or do even the real-time imaging, which is much faster with some photomultipliers. So there's something we can detect. I would like to touch upon one very controversial but very, I would say, mind-stimulating phenomena. And it's a question if now that we know that cells can both absorb and generate light, could they use it for some interaction? So could cells talk using light? So the prototypic phenomena are experiment, I would say, many tens of research groups over the last 100 years tested was this kind of experiment where we had a chemical barrier. If you press next, then this should start to move. There should be this photonic signal moving through, back and forth through the barrier, just showing that if you have the cells separated by the chemical barrier, still the photonic signal can pass through, back and forth, basically. So just showing that while there can be, if the experiment is properly set, and there is barrier for molecules to penetrate through, even excluding the volatile communication. There are some experiments showing that certain cells or parts of the cells, such as mitochondria, we have experts here in the audience on that, can actually trigger the change of the behavior of the neighboring organism or cell culture, although it's perfectly chemically separated. So this is very exciting, and we covered that in several reviews over past 15 years. As Geoffrey mentioned, this is this fantastic book we put together with, I was saying fantastic because I just did a little bit of work. It was mostly by Daniel Fels, who spearheaded the effort. I still like it because there's many interesting ideas there. And last but not least, of the advertisement of our latest book, and next slide, which was also mentioned, where we really put together all these things which I showed in the very beginning regarding the fates of excited states. So really physics, photophysics, photochemistry, and what is known currently in this topic of ultraviolet photon emission, which is just a phenomena for this biological autochemiluminescence. But I don't want to take much longer because we just are going to have the next major speaker coming. So thank you very much.
Thank you very much for that introduction, and it really gets us thinking about what actually happens when the photons impinge upon molecules and biomolecules. Our next speaker is Bob Fosbury. He's an emeritus astronomer at the European Southern Observatory and an honorary professor at the Institute of Ophthalmology at University College London. He is an astronomer who worked for 26 years at the European Space Agency as part of ESA's collaboration with NASA on the Hubble Space Telescope project. Based at the European Southern Observatory near Munich in Germany, Bob joined this initiative in 1985. That was more than 5 years before the launch. During the latter part of this period, Bob served on NASA's ad hoc science working group and ESA's study science team as they developed the instrument concepts for the James Webb Space Telescope, the next generation space observatory. Since his retirement, I don't think you've really retired, Bob, but since his retirement from the ESA in 2010, he has developed interests beyond astrophysics. These led to studies of the interaction of light with biology when, during a fellowship at the Institute of Advanced Study in the University of Durham, he made contact with the visual neuroscientists at the Institute of Ophthalmology at University College London. Some are present on this call today, which led to his honorary position where he works in an interdisciplinary international team to understand the effects of broad spectrum sunlight on metabolism. Bob, I invite you to present your paper.
Thank you very much. And thank you, Michael, for your introduction. I liked your discussion of the structure, which is of great interest to us. Thank you. I'm cognizant of the fact that we have a rather broad audience today, and I just wanted to start off by saying that I know some of you will be wanting to hear about how to design lighting systems and how to design buildings. I will touch upon that topic today, but my intention here is to give a very broad overview of light and life in the universe at large. And when I started this work over 10 years ago with Glenn Geoffrey in London, it was certainly not with the intention of studying astrobiology. But as we've gone on down the road, this looks more and more like astrobiology. And there are some very, very close connections between what we're studying in life and what we see throughout the universe on much larger scales. And that's what I want to draw out of my discussion today. I'm going to start off with a visual abstract just to give you some idea of the path we're taking today. And this is a very simple one, which has a very simple set of words, but it actually contains the essence of what we're doing. What we're saying is life thrives on the starlight that casts its shadow. Now, I chose those words very carefully. I didn't use sunlight, I used starlight, because I think, in fact, the conclusions we're coming to is that the mechanisms we're considering with the interaction between a star in a stellar system and its planets will be common to all planets, at least around similar stars in the universe, i.e. many billions and billions. So this is not a restricted topic in any sense of the word. And we're talking about the interaction of light with the metabolic process as distinct from the interaction of light with photosynthesis. So in fact, what we're proposing is a partner of photosynthesis, where the light is interacting with, principally with the mitochondria and the metabolic process, rather than with generating chemical energy from stellar photons. Now, this is the situation, I think, as most people understand it today. We have a sun, we have plants on the planet Earth. Plants and algae will use sunlight to convert photon energy from the star into chemical energy, sugars, carbohydrates, and so on. And those sugars can be metabolized by other life forms, notably animals and fungi, can be converted into useful energy for cells, ATP. So we have this correct connection between the sun and the plants, and then a connection between the plants and the mitochondria to, in some sense, complete the cycle. What we've noticed and what we're working on is the fact that there's another channel in this diagram, which is, we believe, extremely important. It doesn't work with the same photons that do photosynthesis, which are typically sort of 2 to 3 electron volts. It works with much lower energy photons between, a tenth or even less of an electron volt and about 1.5 electron volts. And there's a very close connection, we realize, between the output of the sun and that lower energy part of the spectrum. Because beyond photosynthesis, these infrared solar photons align very closely, very tight, surprisingly tightly with the metabolic activation energies that drive all of life on the planet. And we believe that it supports both mitochondrial production of ATP and also repair of biochemistry in aging animals and fungi and so on. So it has two purposes. One is to accelerate the rate of ATP production, and the other is to maintain all the repair mechanisms in the body. So that's what we're going to cover today, and we're going to take a very broad view through the whole universe in order to follow this story. And this is basically the plan of the talk. I'm going to talk a little bit about the very first light in the universe after the Big Bang. This may not sound terribly relevant, but it's actually relevant in a rather interesting way. It's now provided a cold universe for us, which enables us to export all our waste heat and entropy away from the planet into the rest of the universe, which is a vital part of the cycle of running life. So it's disposing of waste entropy that's generated by creating the low entropy systems that keep our lives running, our homeostasis. The second light is after the first stars ignite in the universe. That's after a few 100 million years. They operate in a very similar way to life, which I will explain in a moment. I mean, they produce energy. They convert nuclear energy, i.e. Through nuclear fusion processes. They convert hydrogen to helium and so on through the periodic table. And they drive their own form of complexity. And their own form of complexity is indeed the periodic table itself. So when the first stars ignited in the universe, this is something we're looking at with the James Webb Space Telescope at the moment. When those first stars ignited, they were pure hydrogen and helium. There hadn't been any nuclear synthesis to produce heavier elements before. These stars live for very short times, as I'll come on and talk about. But let me move on from that. The third section in the talk is to talk about planets, molecules, and life, and that is the evolution using stardust, starlight, and time. The time that it's taken to evolve our present state of evolution on the Earth is long. It's a third of the age of the universe. And that time has been the essential resource for biological evolution to produce the complexity that we see around ourselves today. And then finally, I'll be talking, this in a sense is the most important part of the talk for this audience, is the sunlight partnerships. That is the relationship between photosynthesis and what we will call photometabolism, which together power complexity and lead to cognition. I know Geoffrey will like this. So that's the plan of the talk. So I'll take a very short slice of time talking about the first light. This is the normal way that cosmologists talk about the history of the universe. We start on the left with the Big Bang and its associated quantum fluctuations. And then there's a very rapid period of inflation called, where it expands in scale by huge, huge factors. About 380,000 years after the Big Bang, the universe suddenly becomes transparent and releases all the photons that it contains. And these photons produce the cosmic microwave background, which we can now observe all around us and has been mapped in great detail. Now, this is the reason I'm talking about this, and that cosmic microwave background started off at a relatively high temperature when it was released, when the photons were released. But it's now gone down to 2.75 kelvins. So it's very, very cold. And this is what we see when we look out through the atmosphere into the sky. We see a temperature which is 2.75 kelvins in general. If we're near a galaxy or another star, it's a little bit warmer, but basically it's a very, very cold temperature. And that's very, very important for this whole process. I already mentioned that about after a few hundreds of millions of years, the first stars started to form. That's of great interest to us astronomers at the moment. And then a long time later, about 4.5 billion years ago, the solar system and the Earth formed. And then we get the first life forming, who knows when exactly, but about 1 billion years later. And then we get the great oxygenation event and the first animals and plants hundreds of millions of years ago. So that's a potted history of the universe. This is the cosmic microwave background as measured by the Planck satellite. That's the third of increasingly complicated satellites that were launched to observe the background. And the fluctuations there, the lumpiness in that, are very, very tiny. They're only about 1 part in 100,000. But they're responsible for the formation of structure in the universe, because when gravitation acted on this huge nebula in the beginning of the universe, the gravity caused higher density regions to collapse more quickly than lower density regions. And this produced the whole layers of structure in the universe, ending up with stars. So these were the triggers of complexity in the universe. Now, apart from those fluctuations, this was the most perfect black body that's ever been measured, as far as I know, at 2.725 and a number of other decimal places after it at the moment. When it was first measured with the first orbiting satellite that measured the microwave background, those were the measurements, and they fit pretty damn well to the Planck function. And interestingly, as an aside, when this radiation was emitted 380,000 years after the Big Bang, it was at a temperature very, very close to 3,000 kelvins, which is about the temperature of a hot quartz halogen bulb, which has some relevance to our discussion further downstream, I would say. And that's one of the reasons I'm mentioning it now. So it's the expansion of the universe since then by about a factor of 1100, which has reduced that temperature to 2.75 degrees. Maybe this is somewhat beyond some people's comfort zone in the audience. I apologize for that. This is deep cosmology, but it's all part of the story. Just to give you a visual impression of what it's like, this patch in the middle is a 3,000 Kelvin quartz halogen lamp illuminating a bit of frosted glass. So you can see the color of it. It's like a tungsten filament light bulb. There's a bit of window light leaching into the room here, and you can see that's quite a lot bluer. The frosted screen is being illuminated by a bit of skylight as well. So that's the main point of that is to say that the universe at the moment is cold, and that cold is very valuable for performing work and allowing entropy to escape. The second light, very relevant for the first stage of production of complexity in the universe, namely the periodic table. These first stars, until a few years ago, a couple of years ago, we didn't know exactly when those started forming. We knew they must have formed within a few 100 million years after the Big Bang, and we surmised that they were massive, extremely hot, and very short-lived. So in fact, they go off like flashbulbs in the universe, and that's why they're actually quite hard to find, because although there are many of them, there are not many of them that are actually shining when we look at them. So you can imagine a dilution because of the narrowness of the time window when they survive. We cannot yet see these individual first stars with the James Webb Space Telescope, but we can see the radiation coming from them through one step, through a two-photon emission process, which I won't try and describe today, but it's another one of these quantum processes when you get a hydrogen atom excited in a singlet state to its first excited state. It can't collapse to the ground state because of selection rules, and it either has to just sit there for a very long time, or it can collapse as two photons. That's a two-photon emission process, which allows you to avoid one of these selection rules. Their UV light and the supernova explosions that result after the end of their lives, after 3 or 4 million years, ionized that hydrogen and cleared the cosmic mists, so that it made the universe transparent to our telescopes. So we can see very early on in the universe through the hydrogen gas, which is ionized, and see the very early structures in the universe. But the important part for this story is that these stars began forging the periodic table, and they do this very, very quickly. The first star that formed in the universe would have gone supernova after 3-4 million years, and it would immediately have ejected a whole range of the lighter elements into the interstellar medium, ready to form new generations of stars. So these first stars forged elements up to iron. Iron is the most bound element, and so you can only get energy from fusion up to fusing to iron. Then they exploded as supernovae, creating these or distributing these, creating and distributing these heavier elements. And the successive stellar generations built the the element distribution we see today. So they built the periodic table. And the sun was formed out of well-populated a periodic table. Now, all this star formation was very active in the early part of the universe. In fact, the rate of star formation, the rate of production of heavy elements peaked about 10 billion years ago. And now it's much reduced. So we're actually living in a part of the universe's lifetime which is much less exciting than it was in the beginning, and is much less productive in terms of producing complexity. But the complexity it produced is now forming its own complexity, as I will illustrate. Now, there's an interesting relationship between the number and quantity of these heavy elements that are formed in stars and the total starlight that's emitted from these nuclear synthetic processes. In fact, symbolically, they're identical. They can be related identically. And if I show this is a toy diagram. This is a cartoon. It's not a real data diagram, but the blue line there shows the increase in entropy in the universe, which is associated with the cumulative production of starlight over the time since the Big Bang, so up to the present time. And the grayed out area shows the rate of star formation in the universe. And that peaks after about 3 to 4 billion years, 10 billion years ago. And it's been dropping ever since. So we see star formation happening all around us, but it was much more active in the earlier part of the universe. Now, this is a cartoon actually collecting the real data on the star formation rate evolution throughout the universe is a very time-consuming and difficult process. And these are the current best estimates of the rate of star formation as a function of either look-back time, which is easy to understand, and redshift. This is the shift in wavelength, which we astronomers use as a proxy for distance, and they go opposite ways round. So we're looking to redshifts of we're looking at look-back times of about 10 billion years after the Big Bang to see the peak of star formation. Don't bother to read the caption at the bottom. Now, this is the sort of diagram of a massive star, a more massive star than the sun. And these more massive stars have a number of shells of boundaries between fusion of different elements. So the whole series of nuclear synthesis processes going on in this star. And the reason I'm showing this is that this star, and our sun indeed, is a low entropy structure in a high entropy universe. So as the stars collapse, when they get dense, they start ordering themselves into a temperature gradient, hotter in the middle and colder on the outside. And then they start nuclear fusion going on in the center, which is an energy source which drives the stellar processes throughout its life. And this can happen simultaneously at different shells, illustrated here. In the sun, it's mostly between hydrogen and helium. But in heavier stars, more massive stars, where most of the heavier elements actually come from, there can be many shells operating at the same time. And this is a low entropy structure. And perhaps I should explain what I mean by entropy in this context. And some people find it quite difficult to understand the concept of entropy. But let me use a quick analogy. Imagine you're a top chef and you're asked to produce a dinner party, a complicated dinner party, a rich dinner party for a lot of people. And you have a very well-equipped kitchen in order to do this. So you go into your kitchen and you produce this wonderfully complex, beautiful meal for all your people at the dinner party. And then you come back the next morning and you have to clear it all up. Well, you started off with a low entropy state with a clean kitchen and some ingredients. You mixed all the ingredients together, made all your pots and pans dirty, and you created a hell of a lot of entropy on the way. And this is the price you have to pay for creating structure. We call it a low entropy structure. you have to provide energy to generate the low entropy structure so as not to break the second law of thermodynamics. And this is what happens. And I just really, for some eye candy, these are some spectra from the James Webb Space Telescope at different redshifts. These are all very high redshifts. So these are all between about 12.5 billion years ago and 13.5 billion years ago. And we've only recently been able to do this because of this magnificent telescope. And you can see in these spectra, I won't describe them in detail, but if you just look at these labels here, we're seeing carbon, we see helium, we see oxygen, we see carbon again, we see oxygen here, we see neon, hydrogen again, hydrogen, oxygen, oxygen, sulfur. So we're seeing the first products of nuclear synthesis in the universe beyond the original hydrogen and helium that was produced in the Big Bang. So this is really, we're observing the origin of the first elements in the universe. And we can now see this in quite significant detail at these very, very early times. It's very exciting for astrophysics. And this is a version of the periodic table, which you've probably not seen before because it's labeled, the color labels are the origins of the different elements in the nucleosynthetic processes in stars. So here we get the blue. These came from Big Bang fusion. So these elements were formed in the first 2 minutes of the Big Bang. After 2 minutes, the universe became too cool to do any more nuclear synthesis. So that had to be done later in stars. But the hydrogen, helium, and tiny bit of lithium were formed in the first 2 minutes of the age of the universe. So all these other colors represent various astronomical processes. Dying low mass stars, the stars like the sun, when they die, they produce the green elements here. Exploding massive stars produce the yellow elements. Human synthesis, this is laboratory things. These don't exist for long enough to be seen in stellar processes. So forget the gray ones, the ones down at the end here. A few elements form not in stars, but in collisions between cosmic rays in interstellar space. So it's quite interesting. Beryllium, if you own an emerald with beryllium in it, the beryllium came from cosmic ray fusion, two cosmic rays bumping into one another. It didn't come from a star. Quite fun. Exploding white dwarfs, these are the cores of sun-like stars, which can explode when they get too massive. They produce the gray elements here. But surprisingly, most of the heavy elements, the very heavy elements, were produced in merging neutron stars. Now that's pretty exotic. You remember in the around about 2015, I think it was, or 16, I can't remember the exact year, when the gravitational wave detectors first found the first merging black holes. This was a real milestone in astrophysics, the first detection of gravitational waves predicted by Einstein from his general theory of relativity. The colliding black holes make significant ripples in space-time. And in 2017, I think it was August, they found they detected a collision which happened at a much higher frequency of oscillation than the black holes. So it was immediately recognizable. And they knew what it was. It was a pair of colliding neutron stars. Within the first half second of that detection, the signal of that detection was transmitted to all the big astronomical telescopes and all the space telescopes. And they started trying to find this. And they did find it as a tiny glowing dot in some distant galaxy. That was quite a task. And the big telescopes were able to follow the fading of this event over several months. In fact, right up till, I think, 2022, when it became too faint to see any further. And you could actually see the emission from gold and platinum and so on from this, where these heavy elements were formed in abundance in the colliding neutron stars. So if you have a gold ring on, it's more than likely that the gold came from a pair of colliding neutron stars rather than an exploding massive star, rather than a supernova. So it makes jewelry much more exciting. So this was quite an achievement to put together this story of where all these elements came from. And the paper that described all these processes was written in 1957. It was published in the Reviews of Modern Physics. It had 107 pages, and it mapped out the whole of the nuclear synthetic processes that operated, neutron capture and so on and so forth, many things. And I happen to know all the authors of this paper, and in fact, I knew 3 of them very, very well. So I was kind of seeped in this story when I was a young postdoc. So this is the complexity that stars produce. They radiate complexity. And I marked in there, just for curiosity, these are the metals, the non-organic elements that you find in enzymes in biology. So this is the connection to biology. And these were formed, these were used at different stages in biological evolution, hydrothermal chemistry kit, and then all the other things. So that's rather fun. Now, I want to draw the relationship between stars and what have I got under here? Stars and life. And I mentioned Schrödinger here because 2 books I found very important in my study of this whole process. The first one is Schrödinger's What is Life, which all biologists should have read. I'm not sure they all have, but they certainly should have read it. Schrödinger had great insights into the relationship between physics and biology. And he would have well understood, and indeed might thought of the stellar system as being a low entropy condensation from a high entropy nebula. And these stars achieve their long-lived stability. I call it a metastability because it's not permanent. But in stars like the sun, it's billions of years. The sun will live for roughly 10 billion years before it dies. And it's that stability is maintained by the balance between the fusion pressure generated by extremely high temperatures in the core, about 16 million degrees kelvins in the core of the sun, and the pressure generated by gravity pushing material from the outside. So that's a temporary stability, okay? And that stability is maintained by the star being able to export its entropy, its waste heat, as starlight. So the starlight is actually the waste entropy from maintaining the stellar stability over a long period of time. And when the fusion fuel in the core of a star is exhausted, the stability fails and the star dies, either in a monster explosion or in a slightly less dramatic way. Now, to me, this is obvious. This is a kind of stellar homeostasis. This is exactly what homeostasis is in a living system. Tell me the difference. Different mechanisms, but we have a low entropy condensation, state of homeostasis. It's driven by energy, food, and it's that food that maintains our life. If we can't maintain the order inside our body, we reach thermal equilibrium with our surroundings, which is, to cut it short, death. And when the fuel is exhausted or we can stop eating the fuel, we die. So there's an incredibly close analogy between the life of a star and the life of an element in the biosphere. Now, just looking at a little bit more detail at this, the stellar death exports newly forged elements into space, which then become available for forming into new stellar systems and new planets and so on. There's the entropy link, which I just discussed. The stars radiate low entropy starlight. The starlight is quite high temperature starlight, so it's relatively low entropy. It can be used again. And that's just like organisms excreting heat and other things. This dynamic kinetic stability in stars endures because there's an energy flow, and that's again akin to life's metabolism. And the outcome is that stars build the heavy elements, the metals and so on, we call them in astronomy, and life builds complex structures through biological evolution and leads to the cognition that allows us to have this talk. So that was a bit of a race, wasn't it? But I hope you get the ideas. So planets, molecules, and life. Now we're moving on from the stars here to what happens on planets. And it's a different process, but it's very, very closely related. These planetary systems form naturally when stars form as a consequence of angular momentum conservation, a very fundamental law of physics. We have to conserve angular momentum. And when the material, the interstellar nebulae containing the gas which will eventually form the star, as that gas collapses, like a ballet dancer, if there's any imbalance in angular momentum of the various components of this collapse, it will cause the star to spin. And all stars spin to some extent because their angular momentum when they were in the nebula added up to something which was big enough to make them spin. And for many stars, they can't retain all the infalling gas because it has too high a angular momentum. And so all stars essentially form with a disk, like a ballet dancer's tutu around them, because that's the material that contains the angular momentum that the star can't grasp. And those protoplanetary disks, as we call them, contain abundant molecular precursors of life. So they're setting the scene for life. And life emerges, as we know, in Goldilocks zones where surfaces can sustain liquid water. And this is the kind of definition of what we know as a Goldilocks zone. It's a balance between the star and the distance of the planet that allows water to be liquid at some place and allow be a substrate for life to form. And of course, we're well aware that on Earth, the primary energy source or nutrient for life is sunlight. Now, just again, to give you a little dip into astronomy, these are images of protoplanetary disks. Now, they look like simulations, don't they? In fact, when I first saw this, I thought, well, it's a nice simulations of protoplanetary disks. They're actually images, but they're images made with a submillimeter telescope. And if you try and take these images in the visible spectrum, the stars are so incredibly bright that you can't see these tiny disks around them unless you have very, very fancy equipment. But in a submillimeter telescope, at submillimeter wavelength, stars are extremely faint. So you can, and the disks are very cold, so you can see the disks. And this is a set of observations of protoplanetary disks made with the Atacama Large Millimeter Array, which is a very big telescope. In the notes to the slides, I've left a couple of URLs so you can look at this telescope, and it's well worth looking at. It's quite spectacular. It's at 5,000 meters on the Andean Altiplano between Bolivia and Chile, and it's a spectacular sight. And you can see the rings formed in these disks. These are rings of place that are cleared by forming planets. So these are planets forming in young stars, and they'll go on. Some of these will go on to be Earth-like planets. So now let's move on to the interaction of the sunlight with the biosphere. The sunlight on the way from the sun, of course, has to get through the atmosphere before it comes into contact with biology. So the atmosphere, the Earth's atmosphere does some atmospheric filtering. Interesting to note that actually the atmosphere is relatively transparent in the visible part of the spectrum. There are very few absorbers, and they're very weak in the atmosphere. And the dominant process of light penetrating the atmosphere is scattering. And the scattering produces the blue sky. That is, a photon coming from the sun to the ground has a good chance of bouncing off a molecule in the atmosphere. And the blue light has a higher chance of doing that than red light. And so we get the blue sky from Rayleigh and aerosol scattering in the atmosphere. At shorter and longer wavelengths, we do get absorption. In the ultraviolet, we get absorption by ozone, very well known for protecting us from damaging ultraviolet radiation. We also get some O2 and O4. I don't know if any of you know what O4 is. It's a temporary collision between two oxygen molecules, and it can generate absorption lines which don't appear in a single oxygen molecule. And it's called a collisionally induced absorption. It's quite important in atmospheric physics. Water, carbon dioxide, methane, nitrogen dioxide are the other dominant absorbers. But it's odd. Well, it's interesting that this difference between the visible spectrum of sunlight and the UV and the infrared on either side is reversed between the atmosphere and biology. In biology, most of the strong chromophores, the strong pigments, are in the visible spectrum. But in biology, in the atmosphere, most of the chromophores are in the infrared and ultraviolet. So you're kind of switching the parity between the atmosphere and the biology. So there's a match, because the atmosphere is relatively clear to reach the biological chromophores, but not so much of it reaches the other wavelengths in biology, which again is very relevant to the story, I think. I only noticed that recently, but it's an odd crossing over of the performance of the atmosphere and biology. The scattering process is rather beautiful, and if you're interested in the atmosphere, it produces many beautiful phenomena. The point is that the scattering process is a way of sorting photons into different colors. We usually think of producing spectra by using prisms and diffraction gratings and so on, but actually you can do it by using a scattering medium. And the atmosphere is a scattering medium, and this is a picture of twilight from the International Space Station. So you can see all the twilight colors here. We're all very familiar with those. But here I've got a fish. Call it Rayleigh's. I've got it here, Rayleigh's fish. It's a little glass fish, which is a very efficient scatterer, a Tyndall scatterer, almost a Rayleigh scatterer, where you shine light through the eye of the fish, and the blue light scatters first, and the longer wavelengths don't scatter until much later. And this is crucial for understanding how light penetrates into bodies, which is the essence of what we're talking about. So the scattering process exploits the fact that you don't have any strong absorbers, and the fact that you can scatter many times, and you can scatter many times, especially for red light. So it's a mechanism which enables you to get red light penetrating deeply into bodies before they get absorbed. So the penetration of light into bodies is allowed by the fact that the absorption is so weak and so rare that you can get the light deep in by multiple scattering. This is just a summary. Since I'm talking about light in general and the light environment, this is a map of the light environment for natural lighting. And it goes here, the irradiance here goes from 10 to the plus 3 up here. This is the unit, this is visible light irradiance, okay? It goes from between 10 to the 2 and 10 to the 3 at the top. And down in astronomical twilight, it's a factor of 10 to the 9 weaker. And the amazing thing is our vision process allows us to see at both ends of this range, which is absolutely remarkable. Very hard to build a detector that will allow you to see a dynamic range of a factor of 10 to the 9. So you see the sources here are sunlight. Whoops, sorry, come back. I went too early. Sunlight, a thermal source. Scattered sunlight, which is again a thermal source. Moonlight, a thermal source. Stars, a thermal source. And the aurora and airglow, which is the only non-thermal source of radiation that we commonly see. Even lightning is mostly blackbody radiation from a temperature of 30,000 degrees, although it does have emission lines as well. So the point is here, this whole dynamic range is filled with thermal lighting. And that's very relevant to what we're going to be talking about. And this is a bit of eye candy just to lighten the load a bit in the middle of the talk. This is more or less the middle of the talk. This shows you a range of natural lighting, the aurora and the Milky Way. Very beautiful picture produced in Latvia. This is a really dark sky. This is 10 to the 9 times fainter than daytime sky. And you see the Milky Way here, but you see this wonderful red and green glow. This is not the aurora, it's the air glow. It's the similar emission process from the aurora, but it's produced by oxygen atoms that are excited during the day by UV sunlight and decay very slowly using some of the mechanisms that Michal Cifra was talking about in his introduction. And they get released during the nighttime. So they produce this wonderful colored glow. Our eyes, unfortunately, because we're using our rods, which are monochromatic, our eyes cannot see the differences in color here, but we can see the brightness of the air glow with our eyes. And this process tells us immediately that our planet is alive, because that oxygen in the atmosphere comes from photosynthesis. So this is a very strong biosignature for a planet. If you get air glow, you've got photosynthesis going on. And this last one, I very much like this picture, very beautiful. This is one of our big telescopes in Chile, the Very Large Telescope. This is the residue of our own protoplanetary disk. Most of that material you saw in the earlier pictures in the young stars has formed into asteroids and lumps of dust and planets and so on. And we're left with the fine dust in our protoplanetary disk, which gives us this lovely Rayleigh scattering disk, which is known as the zodiacal light by amateur astronomers. But you have to go to a dark sky to see these phenomena. Right, moving on to the relationship between sunlight, the sky, and health now. And the next slides I'm going to show illustrate the processes which show us what we gain from sunlight, but also illustrate what we've lost from this by living in a built environment where we have very much restricted the available spectrum that shines on life. So this removal of the sun's extended thermal spectrum, hot spectrum, coming from a hot body, broad spectrum, Planck function spectrum, is seriously impacting public health at the moment. And our goal, Glenn Geoffrey and myself and Scott and Roger Schwelt, is to restore the evolutionary status quo. You know, we talk about lamps some of the time, but we're really trying to get people into sunlight because sunlight is absolutely essential for healthy living. I put this up to show, don't worry too much about the bottom. These are all the absorbers that can absorb light coming through the atmosphere. Concentrate on the top of this diagram. This is the incoming solar radiation. About 70–75% gets transmitted to the ground. And if we move away from the visible near-infrared spectrum to the mid-infrared spectrum, this is where that energy has to go. So it comes into the Earth, gets absorbed by the ground, by the oceans, by life, and so on. It goes through all the dissipation processes that we've been discussing through all these meetings before. It's a dissipation process where that light is used to perform all kinds of biological functions as it goes through and degrades into low energy photons. These low energy photons have to escape through this atmospheric window here, broad atmospheric window at 7 to 10 microns wavelength. And of course, the quality of this window is what we're so concerned about because of global warming. It's global warming which depends on the transparency of that window. And we're changing the transparency of the window. So all these people who are climate skeptics are talking nonsense because they're worried about climate change and they're missing the real problem, which is actually the cause of climate change, which is the fact that we're blocking the atmospheric window that allows light to escape to the cold of outer space. This is linking back to the cold of outer space. We need a cold outer space in order to get rid of all this entropy we've produced by organizing life and so on the planet. this is the representation of the solar spectrum that we're all used to, lighting engineers, biologists, physicists, and so on. So this is units of watts per square meter per nanometer. The black spectrum is the spectrum before it's gone through the atmosphere. The red spectrum is the spectrum where it's gone through the atmosphere at about 45 degrees solar altitude. The blue light is the light that's been scattered out of the sunlight to make the blue sky. And the orange light is just the sum of the red and the blue lights. This is the light that we get to use on the surface of the ground. So I'll only keep the orange light in the subsequent builds of this slide. So this is the solar spectrum as we see it from 200 nanometers to 2000 nanometers. So this is an illustration of what we know this light is doing for biology. It's producing vitamin D right at the atmospheric cutoff down here. Very critical use of the very shortest ultraviolet wavelengths that manage to get through the atmosphere under some circumstances. Photosynthesis going on in the visible, mostly in the red and the blue, and then what we call photometabolism going on throughout the infrared, extending probably well beyond this spectrum. Scott and I have been discussing this in some detail recently. We're not sure how far you go into the infrared and you still get photometabolic absorption of light, which is meaningful. At some point, you degrade to such low photon energy that you get the energy from the temperature of your body, the thermal energy of your body. But in this broad infrared region, we think we're performing positive functions for photometabolism. Now, what happens when we think about the built environment? We move on to this situation where the blue line here, the dark blue line, is proudly presented by the lighting manufacturers as their best-selling window glass for making buildings manageable in a thermal balance sense. So they're restricting the light going into buildings to the extent that you have cut out all of the far red and all of the infrared light. In fact, you see the green line here is a typical white LED, which is also restricted to the visible spectrum. And this dark red line here is actually the transmission of my hand. You'll see my hand later on. Some of you have seen it before, but this is the light that gets deep into biological tissue. And these things are these windows are largely non-overlapping, and this is the problem we're facing. So we've moved into a situation where the only light that we're getting inside buildings is in that narrow blue-colored region of the spectrum. All the other light has been removed from us. And this is really serious, I have to say, for whatever the mechanism is, and we'll be talking about the mechanisms, whatever the mechanism, this reduction in light inside buildings where people spend most of their time these days is very serious. So moving on now, I want to spend a little bit of time talking about vegetation, because vegetation has such a profound effect on the near-infrared light environment. And we know this because we know there are very beneficial effects of getting out into vegetation. Why is it so beneficial? Well, plants actually scatter light beyond 700 nanometers to regulate their own entropy control. Because if all that light entered a leaf and was used to heat it up, they would heat their leaves up too much to be able to perform efficient photosynthesis. So they want to get rid of near-infrared light. They get plenty in their own mitochondria to do photometabolism, but they want to get rid of the light in their leaves because it would heat it up and they'd lose water and so on. So the light longer than 700 nanometers is their way of exporting entropy into the outside world for them and reduce water loss. This creates an incredibly rich near-infrared environment when you're in a vegetated environment. And if you look in a forest understory, you're completely saturated in near-infrared light and you have very little visible light. It's really, really dramatic. And both animals and fungi exploit this resource for all it's worth. And this is an example of how light interacts with biology. This is I don't know if you know, people know a lion's mane mushroom. It's pure white. It's the purest white, a lion's mane mushroom. And what I've done is I put a lion's mane mushroom on the top of a white light source, an incandescent light source, and it glows bright red. And it says, "Look, Mum, no pigment." There's no pigment there. It's just a scattered light. It's sunset light. It's the red light, that's the infrared light and red light, that's able to penetrate into the body of the mushroom. And what it does in mushrooms, in the forest understory especially, it allows the infrared light to penetrate into the storing body, the sporing body in the gills of the mushroom, and it provides the energy required for spore production. And it turns out that this infrared light, we believe, is incredibly important for reproduction processes across life, all life, humans, animals, plants, fungi, the whole of life. The infrared light accelerates the process of metabolism in all these bodies and allows them to generate enough energy to reproduce. This is an infrared picture of a landscape in sunlight. Its peak wavelength of transmission and reception of the camera is around 800 nanometers. And you can see the sky is there's a blue sky, is very dark, and all the vegetation is extremely bright. And this is just plants getting rid of their waste entropy, and it keeps their leaves cool. And both Scott and I did experiments recently where we had a wooden fence illuminated by bright sunlight with leaves from climbing plants projected against the fence and a radiative thermometer. And if you point the radiative thermometer at the fence, it was measuring above 40 degrees. The leaf was around 30 degrees, and the sky actually reads about -50 degrees. So the huge temperature gradients, and there's a real temperature gradient in plants between the leaves and their surroundings because they're exporting their entropy so efficiently by the scattering structures within their leaves. This was a little experiment I did. Actually, I was with Glenn when I showed him this experiment. It blew his mind. This is looking in an understory. It's underneath a whole set of ferns growing in my garden. So I was looking in the dark understory of all these ferns and waving my spectrometer around underneath the ferns. And you can see the whole spectrum here. There's a tiny bit of green light you can see, but look at the near infrared light in the understory. It's incredibly rich in infrared light. And so you can imagine in the forest understory, you get this incredible diversity of animals and plants, all using lots and lots of energy. They're all soaked in near infrared light, and it's incredibly beneficial for them. The little experiment here, just in the red part of the spectrum, the red spectrum is an observed spectrum with that little spectrometer I just held up, and the blue spectrum is a model of what the sky should be. This spectrometer was pointed at the sky from the top of a hill, so it couldn't see any vegetation directly at all. It was looking directly upwards into a clear sky. There were trees and grass around, but the spectrometer couldn't see any vegetation. And this green picture here is the difference between these two spectra. The plants are staining the sky with their excrement. The light, the infrared light they're rejecting from their leaves actually scatters back from the sky, and you can see the vegetation reflection in the sky. A couple of very short movies. These show a rather lovely picture of the Earth from 31 million kilometers, 31 million miles, sorry, where a spacecraft was flying by the Earth. And you can see the continents in brown. That's the moon transiting in front. You can see that was Africa went past, and you can begin to see South America. You can hardly see South America there because it's covered in cloud. So that was just a red, green, blue composite. I'll show it again just quickly. You can see Australia down here, Siberia, Saudi Arabia, Africa, the moon. Now that was one movie made. The same movie had another filter where it was infrared rather than red. So the red channel's been replaced with the infrared filter. Now look at Australia red, Siberia red, Northern Europe red, Sub-Saharan Africa brilliant red, the Amazon brilliant red, and the Northern American forest brilliant red. The planet looks quite different when you look at it in the infrared. There's this dramatic change in the reflectivity of the planet as you move from the red in the infrared. And in astronomy, we call this the chlorophyll red edge. It's where chlorophyll becomes transparent. And we go into this scattering dominated radiative process inside life, where you get the very bright infrared light coming out. Now, I don't have time to make more than just a few remarks about the penetration of near-infrared light into living tissue, but we know that light from 650 nanometers to around 1200 nanometers can penetrate deep into living tissue. In fact, Glenn and we produced a paper in Nature just a few weeks ago that showed the transmission through the thorax from the back to the front. That's very interesting, the fact that it happens. We also think that the absorption and localization of the light before it's absorbed within the body is very much influenced by photonic structures, by the nanostructures in the body, in the way that it's also influenced by nanostructures in leaves and plants. And I just illustrate that by, well, you've seen this picture before. This was a picture from Nature, which I've actually reprocessed to show more clearly. You can see the veins under the chest skin. You can see all the hairs on the chest, obviously a hairy person. This is my hand. You see the veins on the back side because the light is diffusing through the body and casting a shadow of the veins on the surface skin. And I think these vein shadows in this picture will absolutely convince you that we're not seeing scattered light, stray light, when we're looking at the back of the body or the front of the body in sunlight. We've excluded all the stray light, and we are seeing diffuse emission from the light actually traveling through the body. And if we look at a plant here, you see these layered structures, photonic structures in chloroplasts, which are doing two things. They're trapping the light to make maximum use of it for photosynthesis, keeping it there longer, so you can squeeze more photosynthesis out of the process. And they're also localizing the light both geometrically and in wavelength. And Scott perhaps will mention this in his discussion at the end of the series, but he's been doing work on this. It's really, really very remarkable process. This is the endoplasmic reticulum, which surrounds the nucleus of all cells and is very closely associated with mitochondrial function. And you see this is also a layered structure. And the layering here, the numerical layer separation here, is perfect for trapping near-infrared light. So we believe these nanostructures are playing a major role in feeding sunlight into the right part of the body for it to do its performance functions. Now I'll end here. What is the time? Yes, okay. This is, again, a relatively short description of the partnership between photosynthesis and photometabolism. And I think for this audience, or for the quantum biological audience, this is probably the most important part of it. So I'll try and make it clear the story that I'm trying to tell here. The first thing I have to do is to say we can understand this process much more easily if we use a different representation for the spectral irradiance. And this is much more suitable for the biochemistry we're discussing. And it's also the formulation that is generally used in the industry, the solar panel industry, the people who are interested in getting electric voltages out of solar panels. They want to move electrons around. They're interested in the energy, they're interested in photons. So what we do is we convert the irradiance spectrum from power, watts per square meter per unit wavelength, into a new description, which is photons per second, i.e. photon flux per square meter per electron volt. And that transformation has caused enormous confusion within the lighting industry, because the transformation is not trivial. And we've been accused of misleading people by telling lies about the transformations. They think we're cheating by doing these transformations, but it's actually kosher physics. And I've left the numbers in the slides. I won't talk about them now, but if people want to go back and look at the printouts of the slides, you can see what the transformation is. This is the transformation. It includes the factor of lambda cubed, which means it looks very different from the traditional way of looking at the solar spectrum. Now, yeah, before I show you my cartoons about what we're doing, I wanted to introduce this metabolic theory of ecology, which some of you will know about, but not all of you will know about. This is a quite remarkable theory that describes a universal, that's for all life, a universal scaling law between the metabolic rate and body mass, a range of more than 20 orders of magnitude in body scale, in body mass. And understanding this scaling law makes it so much easier to understand the whole process of metabolism and how it determines lifespan, rate of growth, and requirements for molecular repair, and so on. And it's employed here to relate the basal, at rest, metabolic rate of animals to the sunlight intercepted by their body cross-section when they're sitting in the sunlight. So we're looking at the energy that's available to a body over a near-infrared waveband of 1 electron volt centered on 1.75 electron volts. And those numbers will become clear. I did a short review of this book in the latest Guy Foundation monthly, was it quarterly report? Quarterly report? Yeah, that's it. But it's a really interesting read for biologists, I think, to understand what's going on here. So this is what the spectrum looks like. You remember the one I showed previously, which peaked in the visible, where photosynthesis was going on. This one here, the sun outside the atmosphere, has this peak at about 1650 nanometers, which is 1.75 electron volts in energy. And this is a significant excess over the thermal Planck function. This is visible in the previous spectra, but it wasn't very apparent because the difference was so small in the previous representation. But this looks very different. And there's an absolutely remarkable biological coincidence here, in that the reason for this excess in the sun is because of solar physics, which is an esoteric part of solar physics, and that is that the solar atmosphere is more transparent at this wavelength than it is at any other wavelength. And that's because the mechanism in the solar atmosphere that produces the opacity in the atmosphere, the mistiness, is the H- ion. That's a hydrogen atom with an extra electron attached to it. And because the proton is not very good at shielding itself from electrons, it only has one of them, another electron can sneak in and attach to a hydrogen atom to produce the H- ion, and it takes 0.75 electron volts to knock it off. So in the spectrum at shorter wavelengths in the sun, that forms the major part of the opacity of the solar atmosphere. When you have weak opacity, it means you're looking deeper into the stellar atmosphere where the temperature is higher. So it's a hotter part of the sun. And it's kind of interesting to note, I don't know if biologists will react to this, but when you look at the NAD plus transformation to NADH, which is a critical part of the electron transport chain, this H plus and the 2 E minus there, that's the H minus ion effectively. So this is resonance between these two processes. Now, if I go on and build this, this is the light that gets through from the sun. These are the big, broad water absorptions in the atmosphere of the Earth. And this is where photosynthesis occurs down here. And this empty activation energy, I call it, is the fact that if you look at the activation energy in the context of the metabolic theory, all the life forms on the planet average with an activation energy of between 0.6 and 0.7 electron volts. This is the activation energy which you need in order to stop chemistry happening randomly within inside the body. You need to trigger the chemistry in the body to activate, and it's activated with an activation energy of about 0.7 electron volts. So this is the contact between the stars, the astrophysics, all stars like the sun, all stars which are from a little bit hotter than the sun to right down to the coolest stars, they will all have this excess. And that excess is available to biology. And you can see that solar energy, this solar energy excess corresponds to the activation energy of life. Now, this is either a peculiar accident or it's the fact that biology has adapted to this coincidence, to this energy source developed by evolution over the last 4 billion years. I would posit that it's the latter. My cartoon of the electron transport structure in the mitochondria, which is divided into individual steps, presumably to avoid doing too much damage by jumping all at once. If you want to move between two energy levels, if you're standing on the third story of a building and you want to reach the ground, you don't want to jump off the third story. You want to move down until you're at the same level as the ground and walk over. So if you're moving electrons along a chain, you want them to be step from one step to another, which is close together. So it's better to do this through short hops than to do it all at once. And presumably that's why the electron transport chain is done in hops like this. And if you look at the numbers here, it gives you a hint that this is indeed relevant, because a human contains about 10 to the 21, you can check this for me, very roughly, plus or minus a couple of orders of magnitude. It contains about 10 to the 21 electron transport chains. And the photon rate from the sun that reaches a human body is about 10 to the 21 photons per second in this waveband. And this is, again, a representation of metabolic rate upwards here, a high rate at the top and a low rate at the bottom in logarithmic scale, and this activation potential. And this is just a sort of cartoon representation of where you get energy from kinetic energy, i.e. the temperature of your body gives your molecules enough kinetic energy to reach making electron transports at a few tenths of an electron volt. We know that quantum tunneling plays a significant role in the electron transport rate, which allows you to reach out to sort of 0.7, 0.8 electron volts. And if you add photons into this mix, you have all of this blue real estate, which is available for moving electrons around in the body. And that can be for moving electrons faster down the electron transport chain, or doing other things that you need to do by moving electrons. So we're arguing here that the photons are, provided we can get them into the biology, and this is why we had Michael talking at the beginning of this, we have to discuss this. Provided we can get these photons into the body, they can very much add to the process of metabolism. And this is showing where this activation energy sits on this diagram, and it also shows where the solar energy fits. So this quantum tunneling is obviously the Schrödinger equation for quantum tunneling. This is the Boltzmann-Arrhenius equation, which gives us the kinetic energy available for doing this. This combination between kinetic and quantum tunneling allows us to live in the dark. The photons allow us to do much more than just living in the dark. There's another coincidence here, which I'm sure some of you will be familiar with, but it's worth pointing out. And that is if you give an electron 1 electron volt of energy, so you could get 1 electron volt of energy into an electron using a photon, the de Broglie wavelength, the wavelength of that electron is 1 nanometer. And we know that the thickness of the barriers down the electron transport chain are between 10 and 14 angstroms, which is essentially a nanometer and slightly more. And that coincidence of the wavelength with the thickness means that you stand a significant possibility of quantum tunneling through the barriers as well. So I think it's called photon assisted quantum tunneling. Now, coming back to this scaling law, which I mentioned, I'm very close to the end now. Coming back to the scaling law that I mentioned, this is a representation of measured animals and plants over many orders of magnitude in body mass, body rate. And these body rates have been corrected for the body temperature of these different things. You know, we have a different body temperature for a human and a worm or a bird or whatever. So these have all been corrected via this exponential factor here of them. But you see there's this amazing correlation between over many orders of magnitude between body mass and metabolic rate. This is the astrophysical equivalence of this, and it's a very close equivalence. This is a very common diagram used in astrophysics. It's called the Hertzsprung-Russell diagram, or the color magnitude diagram. It's been used to study the structure of evolution of stars for 100 years now. The magnitude, that's the brightness of the star, increasing upwards here. Magnitudes go the wrong way. This is the metabolic rate of the star. That's the rate at which it emits energy. And the color is a measure of the mass of the star. So this main sequence here, where most of these stars sit, is entirely equivalent to this metabolic scaling law for life. The extra bits and pieces here are are stars at the ends of their lives. They move away from this almost linear scaling law. It's not quite linear, but there are lots of details. This is produced by 212,000 stars near the sun using the Gaia astrometric spacecraft. These are very highly accurate points, but you can see this scaling law here. And this is another way of representing the closeness of the analogy between stars and life in terms of their basic physical process and structure. Going back to the metabolic scaling law here, I've made this very simple diagram. I've kept it very simple because this was the diagram I used to discover this just a couple of weeks ago while I was on holiday. I knew about this green line here because this is the scaling law coming from this literature about the metabolic scaling law. So this is a fruit fly down here and a human up here. And this has the scaling law index of 3/4, which was explained by Geoffrey White in his Geoffrey West in his book, which I talk about. What I've done here is simply I've taken the cross-sectional area of a fruit fly, and I've worked out how much solar energy would would intersect that cross-section, and did the same for a human, and just plotted it on this diagram as a 2-point diagram. And I must admit, when I saw that it fell on essentially exactly the same slope, but a factor of about 5 higher up the scale, I was kind of blown away by this, because I believe this telling us that the basal metabolic rate will keep us alive, but by doing more than that fast, we need sunlight. We don't need it all the time, but we need sunlight. So I think this is us thriving in sunlight, and we just survive without sunlight. So in my humble opinion, this is the strongest way of representing the argument that we have that sunlight interacting with the metabolic process in life is essential for allowing us to thrive. I'll finish off with a cartoon which really pulls all this together. I'll start off with our cosmic microwave background on the top left. This is our cold space. When stars begin to be generated, they forge the contents of the periodic table. That's their complexity generation. They eventually forge the sun, which gives us, by virtue of its waste entropy, it gives us photosynthesis, it gives us photometabolism, it allows us to create our own complexity, and we radiate our waste entropy back to the cold of outer space. So we've gone around a cycle here. And you've seen this diagram before. The ability to radiate to outer space is very important. And I would posit this answer to Schrödinger's question, what is life? Life is starlight, stardust, and the cold of outer space. Thank you very much.